All right, so this is the first of a handful of online workshops or think tanks or just discussions and presentations to talk about the science behind a lot of the cures that we're doing. We also have a parallel series of journal clubs that Ellis is primarily leading. He had one last week. When we do the journal clubs, we're going to do those 2 different times. And then the workshops are going to be a little bit more spaced out. They're all going to be recorded, but it's better if we can get them live so people can have some interactions and things like that. All this information is on the mdhcures.org website. The videotape versions of things will show up under the videos, and under meetings and workshops link, there's a calendar where you can find the upcoming events for the entire semester. So we've actually got pretty nicely organized, and we have a lot of really cool and interesting things, and they'll all be available on video form. We're loading all of these up in YouTube so that people can easily access them on any format without passwords and stuff like that. So today we're going to talk about MDH, phosphorylation, and regulation. I'm going to do this kind of a mixed presentation. It's both about the science leading up into why this might be a thing to study as one of your cures, talking about some of the data we have, and then talking about some of the collaborations that we can do towards the end. But I'm also going to be interweaving this in different ways. If you're interested in doing MDH cure that involves phosphorylation, some different points that we've seen on how to maybe incorporate various elements as a cure. I stole a few slides from the last cohort meeting. So if you were in cohort 2023, you saw bits and pieces of this. But either way, it's a nice background for what we're doing. And this is, again, an ongoing workshop presentation that are all going to be online throughout the year. All right, so MDH. Now I'm very human-oriented, mammalian-oriented when I do my research. I've been doing research in lung cancer research for a long, long time. I'm usually very quick to bring out my dissertation from the University of North Dakota to show that I did MDH purification and interactions way the heck. I was the first person to ever do a defense of their dissertation using PowerPoint slides. And that was actually a disaster. Unlike this one, where I have way more slides than I did then. Sorry. So anyway, so what is MDH? Of course, we all know that in mammalian cells, as well as other organisms, there's multiple compartments, multiple organelles, multiple isoforms. In humans, every student knows, the Krebs cycle. They don't pay much attention to gluconeogenesis, where MDH has a different role. And of course, the primary role ascribed to the cytosolic MDH is in the aspartic malate shuttle, or AST shuttle. It's a lot more complicated than that, actually. And so a couple of these slides are going to talk about, why are we studying MDH in a cure? Why will students be interested in MDH as a cure? Because we tell them and we give them grades. Yes. But in actuality, MDH is really very complicated and involved in a lot of different metabolisms. So in diabetes and starvation with gluconeogenesis, we have different directions that are happening with MDH in the cytosol and the mitochondria. Here we see the mitochondria, oxaloacetate malate being shipped out through the aspartate malate shuttle, as we saw before, and then malate going to oxaloacetate through Pepsi K, and off we go to glucose. In adipogenesis, focusing on amino acids that are going to be involved in creating the carbons for doing this, we have MDH going in different directions, both in the cytosol and in the mitochondria. In hypoxia, where glucose is being used at a much higher rate, we see MDH going from oxaloacetate to malate, the opposite direction we saw in adipogenesis, or in starvation and diabetes. And of course, in cancer, especially when you start looking at glutamine metabolism, where amino acids have to supplant the need for respiration because glucose is not able to meet those demands, you see MDH is being involved again in the cytosol and the mitochondria in different directions, and only partially using the TCA cycle and not involving that classic aspartate malate shuttle. So there's a lot of things that MDH is doing. It's a lot busier than we typically think and see in a textbook, and that's kind of eye-opening for students. One thing is the directionality. when people study MDH and they talk about MDH, they look at this as malate to oxal acetate, because that's the directionality we see things in the Krebs cycle. The only problem is, holy cow, it's at standard state, its delta G is pretty positive. So there's got to be something happening, because our Krebs cycle is going, we're making MDH from malate to oxal acetate. So, it's about coupling. It's about increasing malate, decreasing oxal acetate concentration by coupling the product formation of MDH as oxal acetate with the next enzyme that can grab that, citrate synthase, which is very negative delta G. So there's the thermodynamic coupling, but also just driving the concentration of oxalate so low that you can, in relative to the concentration of malate, you can make this a little less positive. But as we see in the next slide, depending upon, or these two panels here in this slide, if you look at cytosol, depending upon the physiological state of the cell, not only does MDH change its direction, here we have the positive delta G, here we have the same positive delta G reaction, but here we have MDH in amino acid metabolism, TCA, going in the, what we consider the thermodynamically favorable direction. I don't really like calling it spontaneous. Things just don't happen. And same thing with the adipogenesis. But what's really interesting is, for these reactions, MDH will have to hand off its carbons to PepsiK or GOT, or from GOT to malic enzyme, and so on. And again, same thing in the mitochondria is happening, different directionality, different potential partners. So this leads to a lot more than the two pathways we see in every biochem textbook that students know. So it's a way to bring in all sorts of different physiology, different metabolism, different outcomes, disease states when you study your cure, when you're studying MDH. Also, of course, there's a good evidence, or beginning of a growing batch of evidence, that MDH is involved in a number of different disease states. I selected 4 here just to kind of focus on. There's a paper you might want to talk to your students where they find a couple of alleles where there's a couple of site-directed mutagenesis that happen right here at alanine 138. There's an alanine to a valine, and that's right in the NAD binding domain. Please talk to your students about conservative amino acid changes, things like this. When that happens, we see changes in expression. We see different of activities in encephalopathy, these patients. So clearly, in a neural disease, we see something. Cancer as well, we see MDH 1 and 2. So cytosolic MDH is classified as MDH 1 in mammalian cells. In yeast cells, cytosolic MDH is MDH 2. Don't ask. Just crazy. And mitochondrial MDH is MDH2 in mammalians, humans. So what's interesting is, a couple of studies are showing that there's modest increases in expression in patients when you look at the RNA transcript for MDH, cytosolic MDH and MDH2 in the normal non-disease adjacent tissue to the disease tissue. When you blot for MDH1 and MDH2, these are control cells here, WI38 MRCs and IMR90s, and we see increased concentrations using the actinic state as the control. You see increases in MDH1 and 2 expression. There's a couple of studies that show that MDH1 is not only amplified, but that amplification of MDH1 is correlates with poor survival progress or prognosis. And then in one study, it was really interesting that when they were looking at tissues, samples in the normal cells and the cancer cells, there was a really strong increase in activity. Now, that increase in activity doesn't really correlate with the modest but significant increases in expression. Something else is going on here. There's evidence for other diseases, Alzheimer's disease. If you look at the central, the CSF fluid in your spinal and around various places in the brain where you're getting the tangles, we see increases in activity and expression. In the right hand too, is they were mimicking some of the issues that happens with Alzheimer's with higher concentration of radicals. And by treating these cells, these tissues with hydrogen peroxide, they saw leading to increase in activity. Does that correlate to expression only or activity? Not known yet. And then of course, diabetes. In a couple of diabetic patients, they found two different alleles changes, a G to an A and a C to a T in different spots. And each case, if you look at the expression of MDH2 in the diabetic cells, in these patients, the expression was lower, the activity was lower. But what's interesting is the stimulation in the diabetic patients, when you add in insulin or when you're changing with glucagon and glucagon-like peptide, you see a diminished increase in the stimulation of metabolism MDH protein. If you just look at general activity, so this is basically an MDH assay where they're looking at HepG2 cells that are expressing the wild type MDH in blue, and then the two other mutations that they found in patients were expressed here. And these aren't huge, changes, well, valine to a methionine and arginine to a cysteine, but you see a decrease in some of the activity. They did not convert that into, detailed kinetic work. So there's lots of things to be done here. And of course, they're looking at what happens to patients with these samples and cells. When you treat with insulin, what happens to the glucose? In each case, in low concentrations and high glucose concentration and high glucose with GLP-1. And you see that the patients with the mutated MDH2, the mitochondrial MDH, have a diminished effect. So what's going on there? Good question. Nobody's gone beyond that so far. So, there's when MDH is dysregulated, there's questions in all sorts of diseases that very few studies are out there. So students should be interested in something like this. So in general, the stuff we're talking about today is MDH post-translational modification, focusing on phosphorylation. There's a lot of areas that people are working in right now, and a lot of areas that are what I'm calling adoption ready. So really, what we're saying is MDH is important. Obviously, MDH seems to be involved in a number of different diseases, but how? What's that mechanism? How is the cell doing this? When there is misregulation, we definitely see some issues. So can we tie this together to drive student learning? When we think something's being phosphorylated, we ask, okay, so what's being phosphorylated? Where is it being phosphorylated? What happens? This is the kinase side of things. And as I'll show you, that MDH is interacting or potentially interacting with other proteins. Does not only this phosphorylation change the structure, function, behavior of the protein, but does it also change the interactions with other proteins? That's maybe a way that nature is modulating metabolism at a level we haven't really studied yet. So what happens to MDH when it's phosphorylated? Structurally, functionally, and then interacting. So we have lots of levels of really cool things to look at. When we're talking about protein interactions, whether, excuse me, it's in the cytosol or in the mitochondria, and we just look at the proteins that share either the product or reactants of MDH2, you see we have aspartate transaminase, we see pyruvate carboxylase or ATP citrate synthase, we see fumarase, we see ATP citrate lyase here, AST, malic enzyme, and Pepsi-K. These are all proteins that are potential interactors with MDH. And in various databases, you can drive your students to, from Uniprot, if you look down at the interactions, they send you to String and a number of other websites where you can have your students then analyze those websites for potential protein interactors. So already you're finding places that you can go in and start to create that hypothesis generation to drive the student cure. So we've known for a long, long time that citrate synthase and mitochondrial MDH interact. We know a little bit about where they interact. We kind of know who's touching who where, but the problem is that depending upon the modeling and who's done the work, and even in one paper where they did cross-linking between purified citrate synthase and malate dehydrogenase from different sources, from bacteria recombinant, from sigma, even from raw tissue, they did the cross-linking and then docking, they got different orientations. So we know they interact. We don't really know exactly where those contacts are. We have a pretty good feel for a number of options. And we don't know what's regulating this, right? There are two models that I can share with you. One, I think, is the best model from one of those published papers. They shared and gave us permission to give the PDB of both citrate synthase and MDH docked with each other. And then one that we did with my students using some of their crosslinks, and we docked them using a different modeling. And so we have two different models, different contacts, some similar, both with a nice delta G of that interaction. And so from this, one of the questions is that we're driving towards, but I think has to go in the back seat for a little bit so we can get a good, robust interaction assay for a student here, is what do these phosphorylations do? Do they drive the interactions? Do they repel those interactions? Do they maybe bias one set of interactions over another? With the docking programs and things like this, I think there's some really interesting cases. Now, in the cytosolic MDH, there's very little information out there except for a lot of predicted models and some big, huge mass spec pull-down studies. But so Chris Bernstein and I have been working together on looking at some of this. So using the Colab AlphaFold, we predicted, we made MDH as a dimer and Pepsi K as a dimer, and then had AlphaFold show us where this docking is. And so there's a lot of cool things we can do, but boy, doesn't that model look nice where you've got part of Pepsi K just looping right into between the dimers of MDH. Where are the active sites on this? Who's touching who? Should we throw this in a hot dock and look at the delta Gs of the interactions and actually both the distance and strengths of these interactions, that you can start highlighting all sorts of interesting potential mutations in the future, right? So again, does phosphorylation drive this? Well, where are the things being phosphorylated? Where are they touching? I've mentioned phosphorylation enough, time to get to the good part. So in a number of different phosphorylation databases, we pulled together, and I'll share with you on our MDH Cares website, what we found so far. And almost monthly, some of this changes. But as of earlier this summer, these are big mass spec pull down experiments where they get tissues, cells under different conditions, some normal cells, some cancer cells, some normal human tissues, some diseased tissues, and then they chop everything up and throw it to the mass spec and say, here's every peptide that's phosphorylated, go to town. I just took somebody's entire career and made it into a 2-second joke. But what it is this nice database where you can look at where proteins have been experimentally determined to be phosphorylated. Now, we've limited the 22 you see identified here for the cytosolic MDH and the 38 for MDH2 are based on a fairly conservative. There's no prediction here, and they've got to have identified in more than one study. So there's actually potentially a lot more. Phosphoserine and threonine, there's some interesting phospho sites that are in common and also that are unique from each other, and as well as the tyrosine sites. But it's not just phosphorylation that's controlling MDH. There's acetylation and methylation. So many different sites of post-translational modification, and the methylation and the acetylation is wide open. One of the nice things is, we'll see in a second, is when you want to study in a purified protein condition, you can use phosphomimics. Well, there are some phosphomimic-like things for both acetylation and methylation you can do. And so I'm going to be focusing on phosphorylation, but there's been a couple of interesting papers looking at methylation on MDH, and again, lots and lots of areas to study. So what I have here is this really ugly map where I've aligned both cytosolic MDH and mitochondrial MDH. And I've got this shared with you, but just briefly, everywhere you see, so on the top, I've blown this up because it's really hard to see. On the top is the mitochondrial MDH, on the bottom is the cytosolic MDH. Everywhere you see a bolded letter, if it's an S, T, or Y, that's a known phosphorylation site on these mass spec studies. If you see an R, then that's one of the important R's in binding the substrate. This is the loop region here in double. This is a loop that closes down and shifts around when substrates bind. Jess Ellis and a whole bunch of folks are looking at that part to see what's going on there. What's interesting is in the cytosol, there's a serine that in mitochondria is a glycine. And what's really interesting is nowhere near there is another charged amine. So is something happening different in this part of the loop in cytosol versus mitochondria? Good question. Wherever you see a black arrow above or below, if it's below, it means that this is a phosphomimic that we've made, validated, and are planning to study or are studying. Same thing for the black arrows above. That means this threonine, which is a negative charge amino acid in the cytosol. This threonine is phosphorylatable. It's been shown to be phosphorylated, and an amino and a mutation we're making. So this is another interesting comparison, things you can talk to your students about. So in the mitochondria, this is a threonine that is sometimes phosphorylated, so it's going to get a negative charge right next to important arginine. But here's a negative charge glutamate, already always negative charge. So this is already kind of a pseudo phosphorylation in the cytosolic. What's happening there, right? Some cool, interesting stuff just happening in the loop, controlling phosphorylation. The blue, green, and later on we'll see some red down here lines. The blue and green are the areas where, through modeling, MDH and CS are known to contact. So these are cross-linked to predicted sites. So wherever you see the green, these are amino acids that are at least close enough to be somewhat contact, which is interesting because that means CS and MDH are interacting right at that loop region. The dark red is where we predicted PepsiK, GOT, malic enzyme, ATP citrate lyase might interact with cytosolic MDH. And this was done by Dave Heck's group using Mo with his community college first-year students at Southwest College, and then also starting to show up with those PyMol collab folds that we've been talking about. So there's a lot of rich information you can look at. You see a lot of threonines and tyrosines and serines that we haven't been near. The bolded Ks, those are lysines that we know are acetylated. So the numbering's crazy on this. So when you look at this, the two sets of numberings, it talks about the variant, whether you have I'm sorry, not the variant, but whether you have the mitochondrial tail attached or not. If it's a bigger number, that's counting from the very first amino acid, with the transit tail getting that protein into the mitochondria still attached. The smaller number means we're counting after that's been cleaved off. So that's more the mature version of it. So these things lead to a lot of interesting things. You can take a tyrosine, turn it to a phenylalanine, take a serine or threonine and turn it to an alanine to take away the hydrogen bonding issues that might be playing a role here. You can use a glutamate or an aspartic acid to mimic phosphoserine and phosphothreonine. There's even a couple papers that are saying that glutamate is giving similar effects to a phosphotyrosine, although good luck getting that through the reviewers, but it still creates a good place to talk about structure, function, intermolecular forces, and important foundational concepts of how proteins fold and interact and then function. So focusing on the site or mitochondrial MDH phosphorylation sites, I've got a model here that'll get started for you. So this is a dimer, and I've only put the phosphorylation in white and pink. White are the ones I'm actually working on with other folks, Amy Springer, Amy Parente, Celeste. And you can see that there are phosphorylation sites all over the place. But what's really interesting is if you do this with a filled model, you see in blue, green, red, white, yellow, black, even a lot of these phosphorylation residues are accessible are accessible at the surface. So having a serine that's phosphorylated buried deep, how does the kinase get in there? Is this an artifact of lysing the cells and things? Well, almost all of these are surface accessible. And remembering that these are studies that multiple people have found these phosphorylation sites. That's a busy molecule with a lot of phosphorylation sites. So there's a lot of cure ideas that you can do without ever touching the lab: bioinformatics, phosphorylation, H++, AlphaFold, docking, all sorts of cool things. MDH is a dimer. And so I have here kind of in lime green, in some weird tan pink, a bunch of the phosphorylation sites kind of in subtle spheres here, but a handful of them that are facing the interface between these two dimers. And there's a number of different regions where they touch, but you can see that these phosphorylations are at or near that interface. So changing those phosphorylations could definitely change how those interactions are happening. again, what's really neat is if you look at the front and back of those 3 amino acids, so this amino acid phosphorylation sites are highlighted in red. These amino acids are highlighted in blue. So not only are they at the interface, but from both the front and the back, their surface accessible. You can see where phosphorylating that could potentially separate or shift those dimers. And Ellis and others, I think Betsy and Chris have been working on how some of these phosphorylation sites alter one active site as well as the other on the dimer. And so they're doing some really cool work on what's happening on the evolution of the amino acids controlling all this. And what we're looking at here is what nature said, aha, let's muck around with these guys to take advantage of those changes that are happening inside the cell. So there's a lot of exciting work that'll show up here as we get going. And then back to the interactions. So those same sites I'm talking about here are highlighted in white, and they're not only between the dimers, but some of these phosphorylation sites are between MDH and citrate synthase. Because we're going to make this publicly available, I'm purposely kind of not labeling things and not showing which amino acids are which, because that's a great student activity in a cure. So here you can see some of the amino acid residues from citrate synthase and the docked MDH. And what happens if you phosphorylate or change this? Is this going to move this loop out where a lot of the interactions are happening? Same thing here, same thing here. So clearly phosphorylation could drive and control some of these interactions. So if we look a little bit more closely at a couple areas, there are some phosphorylation sites like out here where in mitochondria, there's an alanine. In cytosol, there's an alanine. In mitochondria, there's a serine. So we've made this a mitochondria because it's also overlapping some interesting areas and near an interaction site. Here's some phosphorylation happening here. We've got this serine to an aspartic acid highlighted here. This is an interesting area where both cytosolic proteins and citrate synthase are thought to interact again near what is considered a MDH dimer interaction site. Here's the loop region. We kind of already showed that both the cytosolic and mitochondrial proteins seem to interact with that loop in some level. What that does to the loop is yet to be determined. But again, there's some interesting phosphorylations in and around the loop. And what I'm kind of calling a little phosphorylation hotspot here in both mitochondria and cytosol, we see in the K here, the lysines, those are known to cross-link in mitochondria. Nobody's done the study in human cytosolic yet. And so we have, again, a hotspot of interaction, an interaction between the dimers of MDH and a bunch of phosphorylation sites that lie in them. So we've made this, and this from a serine to a threonine. We've been looking at this activity for well, actually, my students made these phosphomimics and chose these 2 years before, I think it was 2018 or 2019. And they were convincing me that these would be really cool because they seem to be similar but different than cytosol. They're in the area where they predicted they would interact. And I said, guys, come on. These things are only one amino acid apart from each other. They're going to do the same thing, and they're probably just going to shut it off. And they convinced me. And I said, fine, do it. And you'll see there's some really interesting results. And so we've been making the cytosolic version of this too over the summer with some of my students. And I'm hoping if you're here, that tomorrow you're in the lab telling us if these things are going to do anything of interest. So what's happening at the active sites, another important and interesting area. This is an old and maybe not totally accurate version that everybody looks at because it's on Wikipedia. But how does this MDH reaction take place? Well, first off, this is very similar to the serine proteases that we see in classic biochemistry lectures. There are 3 amino acids. There's a serine, a histidine, and aspartic acid. And the aspartic acid and histidine in the active sites of MDH and LDH are very similar to that, just without the serine. In this case, the aspartic acid pulls a proton or extracts a proton through its hydrogen bonding, activating the imidazole to start doing a proton transfer between the substrate and the anhydride or the nicotinamide ring there. Arginines are important for holding the substrate and even being part of the reaction. It's more complicated than that. I'm going to have you look over here to the upper right. This is a cytosolic MDH where there's the active site histidine. The aspartic acid is over here somewhere. Here we see serines, isoleucines, glucines, and things all complexing with malinate. So a malate derivative or substitute here. So it's not as simple as just arginines binding, and even serines are binding. But if I take your attention to over here, here is the D, or the aspartic acid, and about 100 or so amino acids away is the histidine. So it's the D and the H that are important in this reaction mechanism. And if I show you, and this is something Mike W is working on, which is kind of cool, is look at the very closely evolution organisms, yeast and humans. We're all very similar, right? They make wine, we drink it. And if you look at around the D here in the human mitochondria, there are 2 threonines that are phosphorylated. In the yeast mito, there are 2 threonines that it's not that they're not phosphorylated, there's just not a lot of data out there. And so around this D in both the mitochondria and the serine are potential threonines, and further down, threonines that are phosphorylated. What's interesting is, I think, is in the mitochondria, there's a threonine sitting out here that doesn't exist in the mitochondria of yeast like it does in human mitochondria, but the cytosolic yeast has a predicted phosphorylation like the mitochondria. So there's a bit of a switch there in the sequence, if you will. When you get closer to that histidine, what's really interesting is C-terminal to that histidine, there is a slug of serines and threonines that are shown to be phosphorylated in human and predicted to be phosphorylated in yeast. So that's what this diagram over here is. So here's the aspartic acid, here's the histidine, and kind of a tan yellow is malate. The arginines are holding and participating in the reaction. But there's a cloud of serines and threonines, and there's a tyrosine sitting over here. Are they shifting these around, or are they participating in the reaction? Are they altering the proton extraction of aspartic acid? Are they supporting or taking it away? Does putting a phosphate here mess around with this imidazole from histidine? Lots of questions. Nobody studied this, right? So there's another area: protein interactions, the loop, this active site, looking at some evolution issues between cyto and mito in humans and yeast. Amy Springer's looking at some of these kind of things in different organisms as well. So there's some really cool bigger pictures here that are not just about human disease. So when you're designing a cure and thinking of the cure, there's the model for creating a hypothesis that Ellis has been doing with the OUR group, and we've been doing with the MCC for a long time. So, what is that background? What information do you need to be able to develop a cure with the kind of information we just showed there? Well, one of the things we did in an earlier recording that, again, is on the mdhcures.org website is the talking of getting a bunch of us together that are interested in this and creating a guide for a protein phosphorylation specific hypothesis development, so that anybody that wants to do one of these has background and kind of a step-by-step guide for the faculty to do this in the teaching lab. So what is the background? Well, there's not a lot of pubs because MDH isn't being phosphorylated, but I did share some data with you that others have published on, shows MDH is getting mucked around in the cells in disease states. We can lean on LDH, which is very close to MDH, and you can see it's regulated, and it's actually targeted drug discovery for anti-cancer therapies. There's a lot of modeling prediction. I just showed you just the tip of the iceberg, and a lot of stuff left to do out there. Looking at folding throughout Fold and Colab, PyMo, virtual reality. Celeste is really, Celeste and a bunch of other folks are doing a really cool job bringing virtual reality to biochemistry and other labs. I would love to see how we could do some modules to build hypotheses using some of these VR tools. And of course, there's it's related to disease and basic evolution. So what does phosphorylation do? What kind of things can you study with your cures in your classes? What is your favorite? What are you comfortable with, right? You can just look at kinetics when you do phosphomimic, look at specific activity, KMV max, like the forward and reverse direction. You can use Arrhenius calculation with some of these. We'll see some of these mutations lead to folding, stability, and instability, KCAT turnover numbers, substrate product competition assays, very classic all enzyme assays using simple spec kind of experiments. You can look at allosteric regulators. I show here the classic PFK fructose bisphosphatase reaction, where different allosteric regulators are enhancing or inhibiting. We know, we teach our students that when PFK and fructose bisphosphatase is phosphorylated, these two enzymes react differently or are allosterically regulated differently to AMP and ATP and things like that. Well, there's your parallel for MDH. Some things are happening there. There are regulators. Structural impact. Do they lose their stability? If you don't have the ability to do a CD heat or a fast thermal melt, simply heating the protein in a tube to different temperatures, spinning down and measuring what's left, the activity that's left, not an Arrhenius assay where you're measuring the activity at different temperatures, but literally heating them for different times at different temperatures, spinning down to see what's left, right? You're looking at stability of the protein, looking at TMs and structure. There's cell physiology. There's some tools coming in play. Sue Ellen, a couple of other folks, our lab, Jess, you're doing some too. We're looking at knocking out CRISPR. Adam Haberman here at USD is working with us to create hex cell lines so we can do some knockout, knock-ins, do some transient transfections, do some kinase hunting, and on and on. There's lots of really cool things you can do with your cure. What are the techniques? Everything. You can keep it simple where you're just doing kinetics after you do the purification. You can do size exclusion chromatography. You can do some thermal melting. These are fairly easy to do experiments. If you want to, eventually we need somebody to really hammer out the interactions by doing pull downs. If you have more background in more biophysical techniques, SPR, mass spec, cross-linking work, doing CRISPR work. So in other words, you don't have to do the really high-end stuff. You can do the straightforward kinetic stuff. But if we collaborate between labs like CRISPR and at JMU and things, there's a wide range of data sets we can bring to bear with these cures. I've talked about a competition assay before. If you're interested in that, love to share. If MDH is actually sharing and shuttling oxaloacetate 2CS, there should be little free OAA around. We'll add an enzyme that will grab a OAA to make aspartic and see if that thing is active. If you use a phosphorylated MDH and there's more OAA, you should see more activity here. It's a fussy little assay, but it's pretty cool. So let's show some actual data that we've got. So purified protein, this thing purifies like gangbusters. These are lower amounts loaded on. It's usually pretty clean. Sometimes you get a couple of high molecular bands, but it's 95, 97% purified. If we just look at the specific activity, the wild type, and some of these phosphomimics in the mitochondrial MDH, we've got several of them that knock down, a couple of them really crazy. Y56T knocks down that activity. You can measure it, but you can't dilute it. A lot of these others, you have to dilute out 1 to 200, 1 to 300, depending on your protein purification techniques. T85 and 240, 224 crank up this activity. 222 inhibits. There's the two that my students said would be really cool to look at. And I said, nope, they're going to do the same thing. And they do the pure opposite. Well, we're now looking at some of the same things in cytosol and their sister residue sites, their homologous sites in cytosol are doing some of the same thing. We got to look at 260, which is right next to here. So here we have a mutant that's activating mildly, one that's really inhibiting, and one that's activating, right? Very cool. Where they lie, how these interact, how these interfere with all these other things we need to study. pH matters, regulation matters. If you look at the pH of the cytosol and the mitochondria, you see that there's different pHs. So what temperature or pH rather you're measuring these activities make a difference, and also putting things in context of the actual physiological concentrations of all these substrates and reactants in the cytoplasm and the mitochondria. Well, there's a cool paper where they show if you mix all four NADH, OAA, NAD, and malate together, the concentrations of the mitochondria versus the cytosol, and change the pH, you see at this ratio, which is similar to mitochondrial conditions, the pH, as it goes up, everybody goes up, but the mitochondrial enzyme is notably higher activity than the cytosolic version. And the flip thing happens if you do things where your concentrations are similar to what you see in the cytosol. Nobody's looked at what happens when we phosphorylate under the mimicking this kind of thing, right? So there's some really cool studies that could be done with the mutants we have. We know this specific activity changes. We don't know, does it look like this, right? What happens to the overall activity at different pHs? Well, with the wild type and two of these mimics, as you get more acidic, the acid, forgive me, we have pH 8, then 7.4, then 6.4 from left to right. pH 8 is kind of the standard pH that we've been testing, and people test MDH for many, many, many years. But two of these mutants, they go down as you get more acidic. Okay. Two of these, it seems to be a very muted response. So is there something happening to some ionizable side chain that phosphorylation is either directly impacting or separating out and changing the pKa locally? And now this one, I've been trying to get a certain student, Andrew and Harrison, if you are here, we need to redo this because this is really weird and highly unexpected, but it looks like, at least initially, that as the pH of the assay conditions decreases, the activity goes up. That doesn't make sense. We need to repeat it, but hey, another cure. So one of the things I talk about when I do cures is, what are these allosteric regulators? What is known? Well, here's a bunch of activators and inhibitors in this list here. And this comes from the website Brenda. Okay, so the Brenda enzyme website, you can click on and find enzymes and their ligands, their role in different diseases. This is basically just showing all sorts of databases and papers. And from there, you can see, that citric acid is inhibiting, and the papers, it'll link to it. Some of them, though, are links to links to links to links and are dead ends. So that's just because it's in Brenda doesn't mean it's guaranteed, but you still have to chase it down. Well, this is a great thing for a student, right? To test that, we took citrate in wild type and increased two different concentrations and controls at 1 millimolar and 10 millimolar. So we're hammering it with citrate, and you see an inhibition. All right, whoops. Pretty much everything's happening. A couple of them might be either activated, being more inhibited than others, but we see citrate still inhibiting regardless of the phosphorylation state. What's interesting is alpha Kg is a known inhibitor of MDH. This mutant doesn't change its activity. If we look at glutamine, no effect in the cytosolic MDH. Glutamate seems to inhibit. You can do some biophysical analysis. This is stuff that Chris Bernson's been doing with us. This is early work. It's actually been developed a lot further than this, where we're able to throw proteins on the beam and look at SACS analysis. And here you can see some shifts when you add citrate or alpha Kg, showing some changes in structure. Now, this might not be something you can do in a semester with your students, but if you get this database, you can certainly share it with them and your students, and then use that to go along with the kinetic data, right? A thermal melt where you're measuring the stability of the protein over temperature. And what's really interesting is what we've seen is the major or the mitochondria has a primarily melts at about 60 degrees, but it seems to be a slight opening or slight melting that happens at about 54 degrees. And depending upon what mutations we use, we see a big shift in the main melting point and a loss of that first part. So we're doing some real structural shifts to it. S45D, Y56, we get two big shifts in temperature, very little changes in the melting with T85, and even 224. So not every mutation is causing the instability of the protein, but some are. And so, this is where we talk about doing Arrhenius equations, heating proteins at different temperatures, centrifuging down and looking how much protein's left, as well as activity, so you can see for structure if you don't have, a 5-color fluorescent plate reader, or a PCR instrument, or a CD that you can use. Doing straightforward size exclusion chromatography, MDH wild type runs down as a dimer. We start seeing this thing glomming together with 222. We see with 224, it breaks its dimer, but then we see some things falling apart as a monomer, which makes sense to us. So maybe there's a dynamic back and forth that's lost when we phosphorylate this, so we get a significant population that's a monomer instead of staying as a dimer. And that is even more advanced when we look at Y56D. These are all experiments that can be done in a number of different ways, including, would be interesting, is just a native gel, right? With all sorts of phosphomimics. We see some more of this bioinformatic or biophysical work where we're looking at, again, some of the sacs. We see changing and unfolding and oligomerization or aggregation for 3 of the phosphomimics. There seems to be some measurable change. This is still real early on work. Chris has been just cranking through some of this work with other students, some of his students, and advancing it. But again, you get different evidence that phosphorylation is doing some interesting things to MDH. So what project tools and resources are happening right now? Well, we've got a big list of cytosolic and mitochondrial MDH phosphomimics that people can use. We have groups of folks that are doing it. When we get done with this, maybe folks that are here, we can identify themselves talking what you're doing. Amy Springer's looking at other organisms. We're doing the small molecule testing, looking at PHCAM, Vmax. We're looking at, and this is a little bit slower project where we're looking at the physical interactions of proteins. There's cross-linking, whether it's glutaraldehyde, formaldehyde, or maybe other protein cross-links. There's some product development that needs to be there. Looking at, I think that the cytosol has got more legs because there's more proteins and less known about it than the mitochondrial interactions. That's where there's a lot of bioinformatic and computational work can be done. Mike Wolniak is looking at that active site and between differences between yeast and human, and also just what's going on in that active site. What if we take those serines and turn it into an alanine? Some of those serines are an alanine naturally in some of these other species. So is the serine OH involved in binding or holding or part of the reaction, or when it's becoming phosphorylation, does it increase that? Don't know. Early on, and I still have them if people want them. I want to get back to that when we get into the interactions, but we took some of the cytosolic sequences and replaced into the mitochondria. So we took out mitochondria sequences and added some cytosolic sequence at different places where they looked like there was contact between CS, or there were some uniquenesses between the mito and the cyto MDH. Some interesting work came out of that, and it's just kind of sitting there if somebody wants to work there. Pymol and VR, we're talking with KP and Celeste and Melanie to do something with both Pymol and a virtual reality, so we can really get a tour, not just learn how to do Pymol or do 3-dimensional work, but get a tour of what MDH looks like, right? When I put on that VR goggle and I saw the loop and the active site, it was amazing. I seen it. You can see the structure. We all can look at the Pymol images, but until you're sitting in the middle of it, you realize how big, and that the perspective changes a lot. I think it can help our students quite a bit. There's a lot of potential phosphorylation sites we haven't targeted yet. There's some interesting SNPs that are out there. Haberman and Peterson, Celeste Peterson are working on a paper to kind of show some of those things. 3 or 4 of us, Jess, myself, Sue Ellen are working on creating, and Adam Haberman are working on creating HEC cell lines. Why HEC? Because you can, there's not a real good liver cell line that I'm aware of that you can transfect easy and work with, but kidney does. And why kidney? Because especially if you're working on cytosolic MDH, you can transfect. You can use all sorts of hormones, insulin, glucagon, epinephrine, norepinephrine, and things like this to get those phosphorylations, get those metabolisms going that happen in liver cells and in the kidney cells. And of course, the biggie question, who's doing what to who, right? That's the question that using these CRISPR cell lines and stuff is, what kinase is phosphorylating when and who and how, and there's going to be multiple phosphorylations happening at one time. So there's a lot of space for, I'm going to stop sharing. There's a lot of space to do research. There's a lot of space to do interesting work with the phosphorylation stuff. And it just is got long legs, and I think it's interesting for students. So, we've got some different folks here that are interested in doing that. I see Ellis. Hi, Ellis. Is in the biochem lab working on stuff. And we're looking at some areas that are kind of really coolly compatible. Yeah, so questions folks have, interested in doing some collaborations with your cures and things, open the floor. Maybe Celeste, you want to kick it off and talk about some VR ideas or what you're thinking, or what are possibilities?
Sure. So this summer at the 2023 workshop, we looked at MDH in VR. And it is really great, especially if the students are already familiar with the protein, and they can recognize some of the architecture of it. Then once you see it in 3D, it really is a totally different rendering for them, that I think is going to be quite impactful. So what we've been looking at is just highlighting the active site, and looking at some of the key residues there. But going forward, I think it would be great to highlight some of these phosphorylation sites that Joe's been talking about. In lab, I've been looking at S8 and at T85 and S45, the surface exposed residues. And so it would be great to highlight that. Also, one of the nice things about VR is you can take these alpha fold structures, like collab fold, and where Joe was talking about predictions of protein-protein interactions, and you can put that into VR. So headset for VR, they run about $300, and the Nanome app for looking at proteins is free. The basic version is free. If you want something a little bit more sophisticated, then you have to pay a little bit more. But I think it's really powerful, and if people want to learn more about it, I'm happy to show you.
How much so what can a free version do, and what can't it do because if you do the free version?
Right, so the free version doesn't give you privacy. So this app is actually developed for drug companies, and so privacy is a big deal for them. But so the free version, you can join public rooms, start public rooms, and people can join those rooms and see what you're doing. It's a little bit weird to have strangers in your room. On the other hand, usually everyone's a scientist and kind of curious. And we're not doing anything that's top secret. The other thing is the free version doesn't give you access to the docking software or the ability to save molecules in the vault.
If you have a model, let's say through AlphaFold Colab that's already folded, or something that's already been published, you can bring them up, you just can't take them apart and put them together.
Oh, that's a good question. I know you can certainly, with the free version, because I have the paid version, I know you can take AlphaFold molecules, the paid one. I don't know if you can do it in the free one. That's a good question. You can certainly download things from the protein database in the free one.
So we're working, I've talked to Jess and Ellis about coming up with a script for KP, and then she would do, like she did with her spike protein, like this cool tour of MDH, the active site, the interface, and all these kind of things. And I would love to then have like a similar version of that with VR for those who have VR. Right? Yep. They can say, after I've seen this, then go play here, go there.
Exactly. Right. I think think that's the way to do it. And like it's really helpful to know the protein first and look at it in 2D on the and Primal and things like that, and then jump to the VR. So that would certainly be doable to make a video that it's where you're basically the tour guide taking people through it. But some of the fun of the VR is for the students, to explore the protein themselves, right? So you can zoom in and you can literally find yourself standing in the active site, or you can, spin it around in your hands, pass it off to a friend in the room. So we also, I think in addition to the tour, we want some room for exploration for them as well.
Yeah.
One of the nice things about the VR too is I think measuring distances is much more intuitive, as well as sort of measuring the angles between molecules. It's much harder to do that on a 2D screen.
Can you measure the distances and stuff with the free version? Do you know?
I think so. I think that's
Okay.
one of the tools you can use. Yeah.
All right. Because I got the free version on some of the VR headsets we have at work, but I have not had time to really see what I can do with it yet.
Yeah, that's always
they
the issue.
installed the free version. I wanted to see what I could do with that before I was like, oh, we really need the paid. So that's what I was trying to.
Yeah, exactly. It's under the tools folder is
Okay.
measuring distances. And the new version of the headsets just came out about a week ago. Those are $500 each, and they have mixed reality. So students actually will be able to go back and forth between talking to you and seeing the real world, and then jumping to the VR world. So they won't necessarily the advantages of that are that you don't have to, like, if you want to have instructions, they don't have to take off their headset to look at instructions, or if you want to interact with them in the real world, they can actually see you.
So Ivy, I sent you a bunch of cytosolic clones tomorrow.
Yes, I saw.
I don't know what to do yet.
It's okay. It's okay.
I mean, that's the whole point, right? These
Yep.
are cytosolic phosphomimics that they've made, but they haven't measured yet. So you'll find out when we do, right? That's pretty cool.
Yeah, it's really cool. Thank you. Yeah.
The cytosolic measurements I showed you was an N of 3. So the one on the cytosol, the mitochondria isn't, but so it'll be really interesting if that keeps going.
Yeah, thank you so much. Yeah, I had one group that just was very adamant that they wanted to do mutations out of nowhere. So I was not ready for it. Thank you so much.
And Jessica Warren's here. That's the Jessica I was talking about that's going to be working on the CRISPR stuff. You want to introduce yourself, Jess?
Sure. Hi, everyone. I'm Jessica. I'm a new assistant professor at Northern State University, which is in Aberdeen, South Dakota. So I'm excited to get some research up and running.
And Tom, have you thought about what you're going to do? Because it'll be next semester, I think, right?
Correct. Yep. My problem is I got lots of things that I want to do. I want to do the pull downs. I want to do some phosphomimic stuff, and I want to do interactions between MDH and carboxykinase. So I'm going to have to focus myself down more.
Well, let's visit when you're ready. And Amy, do you want to talk? You've been doing some interesting things also, not just with the human stuff, but other organisms. Do you want to mention anything?
Well, we haven't got much interesting yet. We've made clones. We've made mutants. We've got protein. Yeah, we're have some phosphomimics in analogous amino acids in the trypanosome MDHs, analogous to the ones in human. And so it's always kind of interesting when those end up in the same place. We don't have any tyrosine phosphorylation in trypanosome, so that tyrosine isn't conserved, it turns out. So we'll hopefully see something soon.
You know, and what's interesting, and I haven't seen any data on it, is histidines in, at least in mammalian cells. We know in plants and in other organisms, histidines are phosphorylated, right? But Tony Hunter and a few others have shown that that's happening in all sorts of proteins in humans. So I guess we might have to eventually add a fourth one to it. Kate, how about you? What are you doing?
Sorry.
Only my name, damn it.
So I actually have those same cyto mutations that you were talking about with the cyto phosphomimics that we made a couple of years ago. So I have the 260 D, 261, and 264. And we had purified those a couple of years ago and gotten some preliminary data. So I'm using those again this semester. And so that's what the students are running enzyme activity assays on right now. We didn't get very much protein for one of them. So I'm not sure how it's going to work out. I'm actually looking at their preliminary data. So that's what I have going on right now. And then we looked at the last time I ran a cure, the PepCK MDH interaction potential site, and we made some mutations in where the Hawk dock had docked PepCK on MDH. And we did some activity assays with that, but we haven't been able to gotten any pull down assays to work. So I was sort of wondering about what possible other options there are for that. And I think that's what I want my students to focus on for their hypothesis module, and then what we might do in the spring in terms of mutations is still that sort of PepCK MDH interaction. But that new doc that you have for the collab AlphaFold stuff, I haven't looked at that yet, so I don't know. And this
Got that
when we
last
did it,
week. We haven't done any.
okay.
We literally
haven't
haven't done anything.
Well, I don't even know. I just Googled what collab was because I was like, what is this? I knew what AlphaFold was, but the stuff we did 2 years ago and the model we have docked was before the structure paper for the cytoplasmic was published. So I was thinking for sure, even with the Hawk dock, we'd want to dock the actual structure, not the model of the.
I think all of it has to be done because I think what you're saying needs to be done. I think we can take the structure then from AlphaFold collab and then separate the two and then put them in the Hawk dock and see if they show up with the same thing, right?
Okay.
Because
I don't know what does this collab do? AlphaFold gives you predictive.
I guess, and I just learned about it just recently and I don't know it very well. Celeste was saying.
I think it just runs the AlphaFold on Google computers instead of taking up a CPU time on your own, right?
Yep. And somebody made a mistake and put like a semicolon in there, and it treated it as two different proteins. And they found out that if you take the amino acid sequence of one semicolon and a second protein, it just separates them and then does folding and then puts them together.
You can make anything bind. You can make anything bind to another protein.
But I think, yeah, it's all about interactions. I went to a conference last summer, and I feel like the theme of the conference was people showing data saying that their biochemistry data predicted, basically supported what CollabFold had found. So I think it's very accurate, is my understanding, which is great.
So it will do like a docking kind of thing.
Yeah.
Is that okay?
And the nice thing is, so you get a crystal structure, you get a structure for PDP, but then you got to clean it up. You got to get rid of the waters. You got to look for and it gets kind of wizardy, right? To make sure it's prepared and then submitted. And what I was talking to Chris Burnson. Okay, I need to get to be a better wizard because I can't just throw junk PDB files together. He said, "Oh, no, do this." And Chris Burnson's actually putting together a module, and there's videos on this to help students do this kind of stuff. And then, again, separate them out, put them in Hawk docs. So you can get that. You can get the delta G measurements and the distances. You could throw it in H++ to see who is close enough where they're in affecting each other. And it just goes on and on, right? That drives a lot of mutations and things, because ultimately I still want to look at interactions and how they're being regulated by phosphorylation. Now, I had a student this summer who did some glutaraldehyde and formaldehyde, and I like those two because they're dirt cheap, right, compared to other cross linkers. But we were hammering it. We were getting oligomers, right? And she was a brand new student, so she hadn't really gotten to pull it down to a size, but Chris has been doing some of the same stuff, and we're seeing hints of it, but we've got to muck around with it. So we don't have a cross link so we can do a pull down. We tried doing pull down, but it's a weaker interaction. Do we need to change the conditions? If you look at the Joy Amini paper out of Nebraska, they show that if you change the pH, you get better changing, add citrate, you get worse, add AT. So they have small molecules that are changing the affinity too. And so maybe we need to do some pull downs with that. I think I'm going to try to, I've got some BC, I think it's BC3, the protein crosslinker, and try to do some crosslink with that. It's a little more expensive, but I think that some of the glutaraldehyde and formaldehyde is actually causing a lysing hydrolysis or something of the part of the protein. Because we see when I add just to MDH, I see a lower band show up and then a higher band. I wonder what that is. Right. And so I think we're getting some fragmentation with the formaldehyde and glutaraldehyde. But that's got to be worked out some more. I think native gels might be an interesting way to go too.
To see like if you can do a gel shift, is that what you're thinking?
Yep. And then, BCE is more expensive, but if it works, then you can cross link and then do all sorts of interesting things, mass spec, pull downs, whatever, just to run a gel and show the shifts. And then do the phosphorylated mutants have a different gel band density? So things going on. If you're interested in doing this, in the Slack workspace, we've got a channel that now just says MDH phosphorylation. Jump in there and say, hey, I'm interested. Maybe take a moment to talk about what you're doing and when you're doing it. If you want collaboration or if you need help with something that we can kind of focus some of the phosphorylation models, we can start sharing the mutations we've got, people that are things that they want to make, so we can kind of see what's happening. That work for folks? So yeah, Slack me. All right. Thanks everybody. Amy, you got a quick second? I'm going to stop recording now.
Thank you.
Sure.